Okay. Welcome everybody to our weekly seminar series at the Department of Marine Geosciences. And today we are very happy and honored to host Dr. Yaroslav Trubin. He's from the University of Haifa and the Geological Survey in Israel. Some words about Yaroslav. He graduated with a diploma in petroleum geology in 2020 from Tyumen in Russia, known as the oil heart of Siberia, where he gained extensive experience in industrial sedimentology and digital geological modeling. In 2024, he completed his PhD at the University of Bonn in Germany under the supervision of Professor Martin Lange. His research focused on Cenozoic foraminifera, including recent materials of proxies for environmental and biogeographical paleoreconstruction. Currently, the postdoctoral research at Geological Survey of Israel and the University of Haifa, under the supervision of Dr. Ori Chaim and Professor Ravital Bachman. The research explores how marine systems and foraminifera assemblage transform under anthropogenic impact and recover from long-term pollution. And actually, Yaroslav is opening a new idea in our seminar of hosting postdocs. So we will have many postdocs giving talks in our seminar. So thank you, Yaroslav. And I leave the podium for your talk and questions at the end. Thank you.
Okay. Yeah, thank you, Nicholas, for introducing me. Okay. Hello, everyone. My name is My name is Yaroslav Trubin. And with this presentation, I would like to show you how you and we can create digital biogeographic models based on only one simple example of giant extinct seas in Eurasia. And before we start, it's very important to note that this is not an independent project. This is part of big collaborative research supported also by UNESCO and performed by scientists from Israel, from Geological Survey of Israel, and from University of Haifa, from Germany, from University of Bonn, and from Tian Shan Geological Society, from Kyrgyzstan. So, and first question of this presentation is, what is giant extinct seas in Eurasia? So, and I hope this map is familiar for you. This is Earth 44 million years ago. This is Middle Eocene. This is Europe, Asia, this is Africa, and this piece of cake is India. This is Atlantic, India, and Pacific Ocean. So, and during Cretaceous and Paleogene, Eastern Europe, Central Asia, and Western Siberia was covered by a huge marine system, so-called Peritatis and Western Siberia. And please pay attention that this is sea, this is not ocean. Peritatis is marine system. This Peritatis is not Tethys Ocean. Peritatis is marine system what covered Eastern Europe and Central Asia. And Tethys Ocean was ocean between Eurasia and Africa. But the size of Peritatis and Western Siberia was like an ocean. Really, it was the biggest marine, shallow water marine system for whole Earth history. And the biggest system provided very important water mass communication from south, from tropical region, to north in Arctic region. And moreover, the system provided moisture for Europe and for Asia. It provided humidity. And when the system disappeared, global climate in Oligocene shifted from greenhouse to ice house, from aridity to humidity. This is a very important place in Earth's history that formed recent oceanography, recent geography, and recent climate. So, and paleontologists focus on question, how did fossil assemblages changed across global changes in this very important place, across KPG boundary, mass dinosaur extinction, Paleocene-Eocene thermal maximum, Paleocene-Eocene mass extinction, Benthic extinction, and the Eocene-Oligocene transition from greenhouse to icehouse. So, and main questions or core challenges of Western Siberia and the Peritatis is biodiversity, biogeography, and the environmental conditions. In other words, who, where, and how inhabited Western Siberian Peritatis. But this meeting is only about biogeography. And within this meeting, we are going to find the answers for three main biogeographical questions. First of all, where was biodiversity hotspots? Second question, how different regions in this huge system connect with each other? And last question, what was the biogeographical role of this huge system? So, and what do we know now about Peritatis and Western Siberia? Main source of information comes from Soviet Union era monographs and very few state-of-art papers in international journals. And main focus of these papers, books, and monographs is taxonomy, no biogeography. But this is not bad, because to create digital biogeographical model, first of all, we need to create a really big database. And to create this big database, we need to open these books, papers, monographs, and try to find images of species and information about distribution of the species. For example, species Nonion layeve was recognized in 2025 by me in Central Asia. And moreover, other scientists recognized the same species in Western Europe, in Paris Basin, in Eastern Europe, and also in Central Asia. So, and pictures provide 100% evidence that different scientists find one same species. So, but sometimes it could be not so simple, because main challenge of identification is synonymy. For example, these two images is one same species, but two scientists named it differently. For example, first scientist called it as Cibicidis mundus, and second scientist named it like Anomalina munda. Of course, word munda and mundus is same but not similar. The word has one root. We can recognize that it could be same species. But for example, most complicated case is these three images. These three images is one species. But first scientist named it as Aster ammonia alaica. Second scientist called it as Discorbis ferganensis. And I, in 2024, call it as ammonia sp. In 2024, I was really fresh and very early career scientist, and I was very careful with naming of the species. Now I'm more sure what kind of the species, but this is challenge. Different scientists call different species with different names. And for example, to check manually all species, one species, I need to spend about 2 or 3 days. And here in Mediterranean Sea, about 200 species. And you can count how many days I can spend to understand biodiversity of your Mediterranean Sea. So, and the result of collecting of database is here on this map. The yellow dots is information what was extracted from literature. The total number of sites is 1,322. And this database is still not completed. And to fill this gap, we did fieldworks. Fieldworks in Western Siberia from 2014 to 2022, in Mangyshlak Peninsula, in Kazakhstan in 2023, in Kyrgyzstan, in Madagen and southern Tian Shan, from 2021 to 2023, and in April of this year in Uzbekistan, in southern part of Uzbekistan. So, and for 10 years, we collected information from 31 sites. This is not a lot, but you can count how many scientists and how many years necessary to collect this very big database. Not one generation of scientists necessary to collect this data. And within this presentation, I'm going to show you results for about more than last 10 years of collecting information and modeling. So, and to provide understanding about biogeography, we collected three key fossil groups, benthic groups, foraminifera, mollusca, and Astrocoda. I believe that everybody is familiar with mollusca. I guess somebody could collect it just for fun across the coast of sea. And if you have this experience, please write it into chat. I'm really curious if somebody has this hobby. And Astrocoda is arthropoda, like a shrimp or crab. And this is microscopic organism, like shrimp or crab, but with valves. Valves like bivalves, but like for crab. And foraminifera is also microscopic organism, but one-cellular organism with a shell. And among these three groups, most useful for paleobiogeography, only foraminifera, because we can find foraminifera in every samples and in every type of marine sediments. Mollusca is too big to be recognized in every samples, and Astrocoda, unfortunately, is not accurate in every type of marine sediments. So I'm going to show you results only for foraminifera. And this is database of foraminifera. And moreover, I'm going to show you the results only for Eocene, because we still have completed data only for Eocene. And we subdivided Eocene into 4 main ages of Eocene. Ypresian here, Lutetian here, Bartonian, and the Priabonian. So, and total number of species is 392. And the number of species from age to age ranges from about 200 and to 130. This is not a lot. This is extremely not a lot. Because, for example, here in Mediterranean Sea, we can find about 200 or 300 species. But this is only one Mediterranean Sea. But these results, 392 species, is from basin like an ocean for million years. This is a very low diversity basin. And probably this low diversity connected with 3 reasons. First reason, preservation status. Yes, unfortunately, everything is still not preserved. And the second reason connected with that Peritatis and Western Siberia was not really friendly for foraminifera. And the last reason, probably we still do not investigate this region. We still do not know everything about this region. We need to do more field works, more collected samples, and we will know more, I believe in it. So, and all foraminifera we subdivided into 5 groups. 2 groups based on a type of shells, wall structure, I mean, and 2 groups is non-endemic and relict. The agglutinated foraminifera is a foraminifera wall of agglutinated foraminifera consists of cement and sand particles. Porcelaneous consists of calcite without pores, and perforadioline consists of calcite with pores. And the non-endemic, the species who distributed not only in Peritatis and Western Siberia, but also in the neighborhood regions. And the relict species who survived across mass extinction, especially this relict taxa, is species who survived across Paleocene-Eocene thermal maximum. So, and before creating model, we can find some trends in this database. For example, agglutinated taxa, non-endemic species and relict species decreased. So, of course, relicted taxa extincted moment by moment. That's the reason why it decreased. But non-endemic taxa decreased probably because the basin isolated moment by moment from age to age. So, and before creating biogeographical model, we need to create paleo-environmental model. And to do it, we need to combine paleoclimate models and paleotopography models. And all the data is available online. And if you want to do your paleoclimate or paleotopography model, you can download this data and play with this data. So, and this black lines is a sea surface temperature. And the lines show how warm water penetrate from Tethys to Tethys and to east coast of Western Siberia, and how cold water from Arctic region penetrated from Arctic to west coast of Western Siberia. And I hope this map is also familiar for you. This is Asia, this is a general map. This is Tarim Basin, this is part of China. Here is Iran. Here is these islands is Cyprus. And Israel, probably first rocks of Israel formed is here, probably, but very, very first. So, and this slide probably is the most important slide for all presentation, because here is explanation how we can create digital biogeographic model. In literature, you can find term species distribution model. So, and to create this model, species distribution model, you need to use Maxent technology. Generally, this is not simple, but generally it works so. You can find on map, these yellow dots on map is places with this species, where the species was found. So, and machine extract information from these points, information about environmental conditions, about depth and about temperature. So, I mean, for example, these yellow dots associated with water depth no more than 200 meters, and temperature is about from 15 degree to probably 8 degree, and machine looks for the same environmental conditions no more than 200 meters and for temperature ranges from 8 degree to 50 degree. So I hope it is understandable. So, and next step is combination of all model for all 392 species. Total number of models is 662 because some species could exist not only one age. And usually I spend about 1 hour to create one model. So, and if you want, you can count how many days I can spend to create all models if I will not eat, sleep, and drink. And you can write it in the chat if you want. So, and of course, combination of all models for all 392 species can show a map of for number of species. And for maps with number of species, we can calculate proportion for agglutinated taxa percentage, for porcelaneous, perforate, non-endemic, and relict species. So, and the first model is a species richness. So the color scale is number of species. And these 4 maps is mapped for 4 main ages of Eocene. This is Ypresian, this is Lutetian, this is Bartonian, and this one is Priabonian. And based on configuration of hotspots, configuration of red and yellow color, we can subdivide these maps into 2 groups. First part of Eocene, first part of Eocene, and second part of Eocene, first and second. because in first part of Eocene, in Eocene and Lutetian, hotspots associated with low latitude. And later, in Bartonian and Priabonian, hotspots shifted to high latitude. Why? Because the boundary of Lutetian and Bartonian connected with closing of Kara Strait. Kara Strait is here. It connected with isolation of Arctic region. So in Eocene and Lutetian, high influence of Arctic region. Therefore, hotspots is associated with a low latitude. Later, no Arctic influence, and the hotspots shifted to high latitude. So the same biogeographical pattern is for agglutinated species, but some agglutinated species could prefer to live in water with Arctic influence. So, and for example, in Western Siberia, before isolation, we can find assemblages with 100% of these agglutinated species. So, this is distribution of porcelaneous species. This is a very unusual distribution because usually in recent shallow water environments, we have to find a lot of porcelaneous taxa. But unfortunately here, just no more than 20% of species. And it provides evidence that Peritatis in Western Siberia was very, very unique biogeographic region. So, and this is distribution of perforate hyaline species. And as we expected, this is highly dominated and highly diverse group, and also controlled by Arctic region. So you can see that the Arctic region dictated everything for paleobiogeography in these regions. So the next one of the most interesting biogeographical pattern is distribution of non-endemic species. So the maps show how species from neighborhood regions, from south and from north, penetrated to Peritatis and Western Siberia. So, and before Arctic isolation, of course, species penetrated from south and from north. Here also from south and from north. We see here a lot of red color. And after Arctic isolations, red color only in southern part. Species penetrated only from south, and Western Siberia formed as a very unique and very endemic place. You can see here a lot of blue color, no more than 20% of non-endemic species. Therefore, 80% of endemic species. It was a very, very endemic place and very unique. So, and this is distribution of pre-Eocenian or relict species. These species show refugium, so places where a species can survive. And for example, this red color associated with deep water environment and with semi-isolated places, for example, like Tarim Basin in China. So, and places where species can survive across across mass extinction could be associated with the central part of Western Siberia, with deep water environment, with Kara Sea, Black Sea, and the Tarim Basin. So this is last one, last model. Local contribution to better diversity. Yes, too complicated, but please don't worry. This is not too complicated like this term. This is just a map of similarity. So blue color shows that every point from this zone is similar to each other. We can find species from here and here and here, everywhere we can find species from this region. But red color shows that this place is very unique. This is transural region. And we can find species from transural region only in transural region, no more. So, and this is, so combination of all information what we have, all models what we created, and all information about distribution of species, let us to create biogeographic regions. Of course, it is very necessary to to create to do geological mapping, of course. But for fundamental science, it's very important to understand when and how biogeographical regions formed. For example, 51 million years ago, Fergana and Tajik Basin was one united biogeographical region. But in 48 million years ago, it splitted, and the Fergana and Tajik Basin is two independent biogeographical regions. So, and cluster analysis is also helpful to understand when two regions could split it. You can find this is Fergana, this is Tajik, and here this is one united region, like one outline of cluster. So, and this is really last one. This is relative probability of occurrences. This map shows where species appeared for 99%. Of course, we can't say for 100% where species could appear. So, for example, this species, Ammonia ferganensis, appeared in margin of Fergana and Tajik basin. So, if we create maps for all species, we can combine it and find most productive place where more species appeared and more species originated. So, and let's conclude step by step. So, first of all, about biodiversity. We created 662 models for 392 species. And to create the models, we used Maxent technology. If you want to do something same, you need to use Maxent technology. Biodiversity hotspots shifted from low latitude to high latitude. And the main drivers was Arctic region, Arctic connection, and Arctic isolation dictated everything. It controlled shifting biodiversity hotspots and all biogeographical patterns into regions. So, and that's all. The maps from the presentation is still not published, but it's submitted to the journal. But now, if you have interest, you can find information about biogeographic setting of Central Asia in this paper, and you can download it here. And if you want to find more information, you can check my ResearchGate or you can contact with me. So that's all. Thank you for your attention. And if you have any question, I would be happy to try to answer you. Thank you.
Thank you very much, Yaroslav. It was really enlightening. I would have loved to be over there with helping you on the field. Looks like an excellent site, all of them. So I open the podium for questions from the people in the audience. Just jump. I don't see all of you, so just go ahead. Question?
Can I ask? My question will be about identification species, more or less how you, for example, how you manage to identify some agglutinated forms, which is, for example, some of them lives in deep, yes, and prefer cool and less carbonate, how to say, less oxygen environment, and some of them like really oxygen and but more rocky beaches, coastline, you know, so more shallow and more deeper. So how you manage this this dividing some species which prefer and that environment and different another opposite environment.
Yeah, thank you for this question. But actually, this is a very complicated story because for recent forums, we can't understand it sometimes, of course, and we need to collect data about environments and about forums and correlate it between each other. Yeah, but if we would discuss fossil forums, this is most complicated question. Yeah, if we can interpret, yeah, data.
I'm sorry. In general, you had samples, yes, from a lot of areas, not a lot of, but and some literature data, and you try to identify fossils, yes, fossil foraminifera, and is it enough 390 and something numbers of species to use this model? Could be it relevant for all fossil, how to say, for all paleo environments? I mean,
Oh, okay. Yeah, sorry, I understand your question. This is also a complicated question because, yes, of course, we took in account only temperature and only paleotopography, bathymetry. If we would add to the model information about oxygen or salinity, the model will be more precise. But this is question not about this question about precising of model. Yeah. But unfortunately, we still do not have information about for all salinity, for all oxygen, and we can create model with this information. When we will have it, we will take it into account. I hope I answered your question. No?
Thank you for question because it's really nice topic. Your presentation was really nice, and I have a lot of questions, and I want to contact with you later maybe because sorry.
Yeah, of course, please, Alexandra. I would be happy to discuss it with you.
Oh, can I ask also one question? I was in Crimea, and I know that it was study field from university, and for us it was like a outcrop with nummulites. Yes, and it's like a pattern for Eocene, like nummulites. It's one cell's organism form. Yes, but they are really big in that time. And did you account these forums in your model? Because it's really how to say, I know that in Egypt there is in some and they are really how to say the how to say they are
State in Russian.
разнообразие,
Diversity, yeah.
sorry, biodiversity were in Eocene, exactly. So, and they're also where they live, it's a really distinct environment, and maybe there are, you don't need a lot of forums, yes, you just need to forums which live in precise, salinity, oxygen, and blah, blah, blah. So to correct and to verify your model to more,
Exactly. I understand you. Yeah. Yes. Thank you.
Because nummulites, they are a really, I don't know, famous foraminifera, yes. And my question was also, did you account these forums or data about nummulites in your model?
Yes, this is also a very good question. And short question, no, we didn't took nummulites because
I will give you.
Okay. Yeah. But because we did model only for small benthic foraminifera, for large benthic foraminifera, it could be one more additional work because there's absolutely another group, and unfortunately, database for large benthic foraminifera is still not created. And this is a big topic, what is discussed in the scientific community now.
Thank you, Yaroslav. Your presentation was really beautiful. Thank you.
Thank you. Thank you.
More question?
Can I ask?
Please.
Hi, Yaroslav. Thank you. It was very, very interesting in the talk. I would like to ask, you said at first that the Perithecus is poorly diverse in relation to today's Mediterranean. Could it be, firstly, maybe you can say if you know from other oceans or marine environments, is it still poorly diverse, or you only took it as an example for Mediterranean marine environment? And second question, could it be that some species did not preserve fine, so you cannot see them in the record?
Yeah, thank you for the questions. Yeah, this is also very important. And first of all, yes, I believe that every species is not preserved in this sediment. And probably these maps, what I showed you, this is map of not real biodiversity in past, this is map of preservation status. Probably the map is about it, approximately. So, and first question is about comparing of biodiversity. If we would compare it with islands in Pacific Ocean, in islands of Pacific Ocean, we can find 350 species, but only in small shallow water islands. Here in Perithecus and Western Siberia, we can find in one point maximum 85 species. This is maximum. And in one point in Pacific Ocean, 350. I hope I answered your question.
Yes, okay, thank you.
Revital, please.
Yeah, I wanted to ask you, you see a very significant change between the early part of the Eocene and the later part of the Eocene. Is it something that is seen globally, or is it something regional or even local?
Now we have a data only for this region. And of course, now we can say that this biogeographical pattern is only for this region. Yeah, but closing of Kara Strait, Arctic isolation, influence on climate, and probably it was first step to shift climate from greenhouse to icehouse. And last step was closing of Turgai Strait and disappearing of all this basin. Probably this basin, two big basins, controlled everything of climate of Earth, probably.
So you think it was that what was happening here had an influence on the entire globe?
Yes, probably. This is hypothesis. Yeah, but I believe that we can find more evidences about it.
Yeah, I guess if you go to the sediment core, the IODP sediment cores in the ocean to that time period, maybe it will be possible to see the global shifts.
Yeah. I believe that we can find the answer to a question in modeling of currencies, because it could work like Gulf Strait, current Gulf Strait in Atlantic Ocean, and it worked so the same, and it controlled climate. So I think so.
Thank you very much. It was a beautiful presentation, and also very important, I think, because we mostly know about other parts of the world, and this part of Russia and the inner Asia is less known. So I think that's really nice and novel, new information. Yeah.
Yeah, thank you.
Okay, thank you very much. If there are no questions, I think we should close. There are questions?
Yes, I have a question. Yeah, Yaroslav, thank you very much for your talk. It was very interesting. I'm not specialist in your field, but I have one question about model technique. I suppose that your modeling based on differential equations. And could you tell some words about it? And could you evaluate the accuracy of your modeling?
Yeah, that's also a good question, but I have to say that I'm not expert in math or programming. Yeah, and yes, this model works like a probability. It shows where is more probable species distributed. Yeah, and we can cut the percent of this probability and find the best percent of probability, and based on this we create species distributing. I'm not sure that it was enough clear. Is it clear or not?
Yeah.
Okay, thank you.
May I ask last question, please? How many samples did you have for this model? Species, but samples.
Yeah, strokes in Excel table is this number plus with this number, 1322 plus 31. This is number of stations, number of samples.
Thank you.
So, more questions? We're still have time, leave it. Okay, so thank you very much, Yaroslav.
Thank you.
Thank you.
Generally, definitely created a lot of discussions here, so thank you very much. And thank you, everybody, and see you next week.
Thank you. Bye.
Thank you.
Thank you. Bye-bye.